All right, thank you. So one disclosure I have is that I work at this company called Airlines. We're working on a medical device, a hearing aid, and we have like 50 patents and so on and so forth, but there it is. But I'm not going to talk about any of that stuff today. What I'm going to talk about is ongoing work we've been doing on developing anatomically based 3D computational models of the middle ear and the inner ear. And this is sort of like an overview slide, and then I'll go into the details of some of these projects that are related to this overview. So we use biomechanics, and that's modeling. And we use physiological measurements, like measure pressures, and we have a 3D laser vibrometer that we can make measurements of. We do imaging, micro-CT, and 2-photon to develop our models and put input into it. And then we develop models of the normal ear, pathological ears, and then we could do surgical simulations and do possibly prosthesis designs as well. So I think it's really a nice synergy between the School of Engineering and the otolaryngology department. And Charles, I think I produced this slide about 10 years ago. The only thing that's changed is some of the details, and the logos, right? So we've been working on these for a while. So let's talk about the specific projects that we've been working on in our group. So one of the things that we work on is the middle ear. One of the unique things about the middle ear, the mammalian middle ear, is that it's got this 4-layer eardrum structure, and it's got 3 middle ear bones. And it's not very clear what the reasons for these is. So it's very scientific in our approach to try to understand these mechanisms. And people have all kinds of ideas. So we take these images, like this is a 3D reconstruction from a micro-CT. It's not an artistic drawing. It's actually from CT imaging. And then we develop models from that. And we can address some of these questions. And some of the 3 basic ideas about what role these play is that it's for mammals are unique in their ability to hear high frequencies. So these structures must have something to do with that. The other possibility is that they have 2 muscles as opposed to other animals that have a single muscle. So maybe these 3 ossicles are needed for independent muscle control of the bones. And another possibility is, Dick Goodall always likes to say that the designer is a safety engineer. So there's a way to keep static pressure from getting in. And Huttenbrink and other people have worked on these ideas. So we have a pretty good group in terms of doing research on this. Hang Shui Kai has been working with us. He's a research associate. Ryan is an MD PhD student. He worked on some of the mechanics, measurements, of 3D measurements of the ossicles. Peter Gottlieb is a new graduate student, and Corey, she's a new graduate student. And we've had lots of projects in the past with Dick and Nick Blevins. And in fact, Jerry's going to talk about one of those projects after me, in fact. Okay, so the other project we work on is cochlear mechanics. And there's these thousands of outer hair cells, and how do these outer hair cells work together to give the cochlea its exquisite sensitivity and frequency selectivity? And that's one of the fundamental questions that's still not known. And so one of the ideas we've had about this is this feed forward, feed backward idea. And the idea here is that the thought is that when the cilia moves due to shearing motion, it produces a force that's more apical, and that's a feed forward of force. So the energy accumulates, and then when the hair cell expands, it pulls the hair cell through the phalangeal process in the backward direction. So those 2 interact to produce this feed forward, feed backward. And we've tested these ideas on simple computational models, and now we want to do it in 3D models. And we sort of switched over to the mouse model because all the nice mutations that are available where the mechanics are well characterized. And John Oglai has this nice mouse model. So working on characterizing the morphometry of that. And Jar Soons, who's a postdoc from Antwerp. He's in our lab, and he's going to develop a finite element model in 3D to test this. The other thing we are looking at is measuring stimulus frequency autoacoustic emissions to inform our models whether they're correct or not, and test these different ideas. And so we've already been working with Tony and John Oglai on this, and we're looking for some help, and you can have your name here if you work with us. This is advertisement, right? So stimulus frequency autoacoustic emissions is one area we definitely need to make more measurements of. So this is nice because this project just got renewed. It's another one that just got really renewed this year. So we're just getting started on a lot of these measurements and modeling studies. Okay, the other project we're working on with Tony is on this lipid model of transduction. And there's a poster here. Jichul Kim's working on that. And we're working on the outer hair cell force generation model, or thinking about doing that, because those models then go back to the feed forward, feed backward models in order to test more realistically force generation in the overall system level model. So again, we're looking for somebody to help us work on this modeling studies. Okay, so the main thing I'm going to talk about today, there's an overview of some of those projects. The thing that I want to talk to you about is bone conduction hearing, and I'll go into a little more detail than I have in the other projects that I've described. So bone conduction has been known for a long time. It's you hear through air conduction, and there's an alternate mechanism for hearing that's through bone conduction. Well, and it's used clinically a lot to figure out whether you have a conductive hearing loss or a sensorineural hearing loss. But the mechanisms for bone conduction are not really very well understood. So there are at least 5 different dominant mechanisms that actually Tondorf talked about, and Stenfeld, when he was here in Good's lab, talked about. And of those, I'm going to talk about 3, which are the cochlear compression. The idea is that when you vibrate the skull, you're doing 3 things. One of them is to compress the cochlea overall, and that allows the basal membrane to move. Another is that you're shaking the skull, and therefore there's an inertial fluid component. And the third is this inertial ossicular chain component. So all 3 of these mechanisms interact with each other. And one of the nice things about these is that you can study these because you can change the degree to which these are in effect under different pathological conditions. And 2 of those conditions are otosclerosis, that I'll talk about, and semicircular canal dehiscence. Okay, so the way we do it is that we develop a model, a finite element model. You take a temporal bone, scan it, this is sort of the size of a refrigerator, put a temporal bone core in the scanner. This is a cross section. You take stacks of these, stack them up, and do 3D reconstruction. And from those, you can develop a model. Then you put in mechanical properties like Young's modulus, density, and so on. And then you have boundary conditions. Then you can simulate the motions, essentially. Okay? So the first thing we could do is, for air conduction, so you put sound in the ear canal, right? And then that produces motion of the ossicles and stapes, and goes down and vibrates the basal membrane. Of course, this is a highly amplified motion, right? So this is air conduction, and we can calculate like an effective hearing loss in this model by doing it the normal way, and then doing some alteration like a dehiscence or fixation of the footplate, and looking at the change in the basal membrane motion, right? So this is all a passive model. There's no active mechanisms in this model. Okay, the next thing we do is do bone conduction excitation, and you shake the entire structure, okay, include that surrounds it, and then you calculate the basal membrane motion again, and then you do it for dehiscence in the semicircular canal or fixation of the footplate, and you calculate effective hearing loss. Okay, so the first thing we thought of is, okay, so do the otosclerosis condition, and one of the things I've been very interested in for years is trying to understand CARHART'S notch. There's this dip in bone conduction in the middle 2 kilohertz range. All the clinical folks know this, but and so it's been known since, I mean, Carhart described this about 50 years, well, 60 years ago. And in fact, I was at Northwestern a couple of weeks ago where Carhart was, and I asked people there, does anybody understand the mechanism for Carhart's notch? And everybody says nobody really knows. So we've been working on this for a while, and I think we got some sort of a breakthrough recently. So the first thing people think of is this inertial mechanism. So you take the entire structures, the middle ear and the cochlea, and you just inertially move it, like I showed you in the picture before. And if you do that, you get sort of a similar behavior. So here's Carhart. This is hearing level due to bone conduction, right? So there's no hearing loss due to otosclerosis at the lower frequencies, but there's this characteristic notch, and then it goes back up. This is Carhart's original data. There's lots of data since then that shows that it goes back up. So when you do inertial mechanism, however, you see this kind of a dip in the hearing loss in bone conduction, but this low frequency is not explained, right? So there's something not right about the inertial mechanism. So then we thought, okay, what about the compressional mechanism? So you apply compression to the cochlea, and you see that there's not much of a change in bone conduction hearing due to compressional mechanism. So that doesn't seem to do it, except at low frequency, it's not, there's no hearing loss produced. So maybe what you need is a combination of the two. And we did that, and it turns out that seems to actually produce this characteristic notch. Now, the different graphs here are due to shaking the bone in different directions. So we don't know exactly what direction all the structures are vibrated in. So we essentially apply the stimulus in different directions just to see if there's a directional dependence. And there seems to be some directional dependence, but there's no data to test this idea. But generally, what we see is there is a notch. And one of the ways we know that this is due to the mixing of the two is that we don't know the phase relationship between the inertial and compressional mechanism. So we actually change the phase, for example, by 90 degrees, and you can see that the notch changes. So the two are interacting in some way. And so I think this is a reasonable start to understand this Carhart's notch. And we just found this out like 3 months ago, and there's a poster, by the way, between Tony and Stefan's lab, so you can see the details of that. Okay, so the next thing is the semicircular canal dehiscence. So basically, dehiscence is essentially like a hole in the superior. It could be any one of the canals, but where it was discovered was in the superior canal. And this was discovered by our current dean, Lloyd Minor. We didn't start this after he got here. Okay, we've been doing this for more than a year. So there's a hole in here, and that, there's lots of things that happen as a result. People get dizzy and have oscillopsia and other kinds of things. But audiometrically, there's a decrease in air conduction sensitivity, and that's due to basically that the energy gets shunted from the cochlea to this pathway, and so you decrease air conduction. But it turns out that there's a hypersensitivity to bone conduction as well as a result of this. So, but there's data that of the airborne gap by Minor's group when he was in Baltimore on individuals and means, and our model is actually pretty consistent with the calculations of the airborne gap in this model, and that with this dehiscence. And we could do all kinds of things with that. For example, we could look at the effect of the location of dehiscence and the size of the dehiscence, which is something we're working on as well. Okay. All right. So basically, as you can see, we combine biomechanics and physiology and imaging to come up with models of the cochlea and inner ear. And we've been collaborating with each other for a while, and it's been a really good relationship. This is some pictures from our group. There's Charles. This is Corey, a recent student. There's Jerry. He never misses a good party. Hang Shui. This is Jichil, who's working on the stereocilia model. Joris, Peter Gottlieb. We have a Head Start program for early recruitment. And this is some of our past members. This is Ryan Jackson. He's just finishing up in my group.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.